All right, here is that second part of chapter 23 that we were talking about. And so this is where we left off, talking about altering gene number or position. Your chromosomal mutations that delete, disrupt, or rearrange any of the loci, that's where the location are, those are usually harmful. This is something I'm not going to go over. Feel free to review the term frame shift mutation. You're going to find that in chapter 16 of your Bio 111 book, your Pearson book. It's the same one. And so your frame shift mutations can be caused by nucleotide deletions and nucleotide insertions, and they can lead to missense or nonsense mutations. So please review this when we're talking about those genetic mutations that can be damaging. And so duplications of small pieces of DNA, those are usually less harmful. So small portions of DNA. The whole point of DNA is to code to make proteins. So if you have repeat sections of that DNA, what that does is it usually just causes a repeat to make more of those proteins. Okay, so they're usually less harmful. And so duplicated genes can actually take on new functions and further mutate. And so an example of this is an ancestral odor detecting gene that has been duplicated many times. And so humans have 380 copies of the gene, while mice have 1,200 copies of the odor detecting gene. So that has helped the mice to evolve a greater function than humans' olfactory abilities. So rapid reproduction. And so mutation rates are low in animals and plants. And the average is about one mutation in every 100,000 genes per generation. And so those mutation rates are often lower in prokaryotes, but the fact that prokaryotes have a shorter generation, meaning that they go from one prokaryote to two prokaryotes to four prokaryotes. So every generation time, prokaryotes have anywhere between a 20-minute generation time to maybe an hour. So your bacteria, your archaea, they are going to reproduce quicker. So even though between one generation to the next, even though they have a lower mutation rate, because they have a higher reproduction rate, those mutations will actually accumulate more quickly. And so that is why your bacteria, you talk about why they mutate so quickly. That is the reason why. And so your viruses have both a high mutation rate and short generation times. So that is why your viruses will mutate so much more than everything else, your eukaryotes, your prokaryotes. So this is just a review of sexual reproduction. Please review your chapter 13 in your Bio 111 chapter, Bio 111 book. In organisms that reproduce sexually, most of the genetic variation results from recombination of alleles, i.e. crossing over. And that takes place during meiotic division. Your sexual reproduction can shuffle existing alleles into new combinations. So the genetic variation happens during meiosis, the making of the eggs, the making of the sperm. But when the egg and the sperm come together, you only get specific eggs with specific alleles and specific sperm with specific alleles. If for some reason a different egg was ovulated that month, you'd get a whole different set of alleles probably. And so this leads to further genetic variation. And so 3 mechanisms that cause that genetic variation, the crossing over, what I talked to you about to review in chapter 13, independent assortment. You're also going to see that in your chapter 13, and you will see independent assortment in your chapter 15. and fertilization. So those are going to lead to great amounts of genetic variation. So that's important because genetic variation is required for a population to evolve. It doesn't guarantee a population will evolve, but without genetic variation, a population will not evolve. And so one or more factors that cause evolution must be at work in order for populations to evolve. And so let's talk about what a population is. Just a little quick review of a population. So a population is a localized group of individuals capable of interbreeding and producing fertile offspring. So to be specific, it doesn't say it here, but when we talk about localized individuals, we're talking about individuals of the same species, and they're in the same area, and they can interbreed, okay? And not only do that, they end up having offspring that they will eventually be able to interbreed. So populations are not always geographically isolated, but individuals of that population typically only breed with members of their own population. So what that means is there's not always a fence separating populations. Now, don't get me wrong. Sometimes there's a fence separating those populations. We talked about this in class about the fences that go up and down I-25, but not always is there a fence, okay? But populations, individuals of a population, they can be kind of snooty sometimes. And so they don't always let outsiders in. So usually you have populations that will aggregate, and then you have some individuals that try to come into the population and mate with the women. And then the males of those populations are like, get out. You're not mating with my women. So that's what that's trying to say. Think about guerrilla groups. Your guerrilla groups, they are not geographically isolated. They can intermix with other guerrilla groups, but it's discouraged by the silverback. He does not like to share his women. And he does not like to share his women with his sons either, or his nephews. And so when it comes time for those sons and nephews to become their own silverbacks, they usually have to get out, okay? There are situations that they're finding that guerrilla groups have several 2 to 3 silverbacks within one group, and there's always one primary silverback and the other ones are helpers. But if they're thinking that they're going to breed, they're going to have to go find their own group. Usually, don't get me wrong, there's sneak mating. Sneak mating happens. So, alrighty. So this is just an example of a crossover of 2 populations where they share a similar land. You got your porcupine herd of your, these look like caribou. So you got your porcupine herd down here. You have your 40-mile herd. And then they have their own general separation of land and range, their range, but you can see there's a crossover of this range right here. And what we're looking at a map of Alaska and Canada. And so, like I said, they're not always separated by a geographical separation, but sometimes they just tend to separate themselves from other herds. So let's talk about gene pools. A gene pool consists of all the alleles for a loci in a population. So let's back it up. When we talk about this, this herd of caribou right here, so of all of the individuals in this population of caribou, they have each individually have their own genetics, okay? In their cells, they have their own chromosomes. Within the chromosomes, they have their own alleles. So when we're talking about a gene pool, we are talking about all of their alleles of every single individual in a gene pool, in a possibility of coming together to make different genetic combinations. And so A locus, we've discussed locus in Bio 111. A locus is fixed if all individuals in a population are homozygous for the same allele. Meaning, if all the individuals have the genetics to have fuzzy antlers, then that's going to be a fixed phenotype in the population to have fuzzy antlers. But you might have something that doesn't have fuzzy antlers. Well, then it's not fixed in the population. So if there are two or more alleles for a locus, locus is the location in which that allele is found. If there are two or more alleles, diploid individuals may either be homozygous or they might be heterozygous. All right, well, let's go ahead and stick with the fuzzy antlers. Let's back it up. Let's pretend all of these guys have fuzzy antlers, and fuzzy antlers is dominant. It is a dominant trait, okay? So all of these guys with fuzzy antlers being a dominant trait, it could be that they have the homozygous trait for being fuzzy, meaning they got one allele from their mom to have fuzzy antlers and one allele from their dad to have fuzzy antlers. That's homozygous, specifically homozygous dominant. Or they could be heterozygous, meaning, I don't know, this guy right here, he doesn't look like he's all that fuzzy. He looks kind of fuzzy. Maybe I'll say he's heterozygous, meaning that maybe his dad gave him an allele for fuzzy antlers, but his mom gave him an allele for smooth antlers. Okay, so with that, that is what it means to be heterozygous. Alrighty, so this right here, I'll just be honest, it's going to be the scariest thing that you do this semester. And once you do it and get it, you're going to realize it's not so darn scary. And it is the Hardy-Weinberg equation. So the Hardy-Weinberg equation can be used to test whether evolution is occurring in a population. It is also used to determine the percentage of a population carrying a specific allele. Meaning, going back to the fuzzy antlers, suppose we want to know how many alleles in the population are fuzzy antler alleles and how many alleles in the population are smooth antler alleles. You could figure out a percentage of fuzzy antler alleles versus smooth antler alleles within a population. And so the Hardy-Weinberg equation, it's going to describe the genetic makeup of a population and whether or not that population is evolving at that particular locus, that location on the allele. So if the observed genetic makeup differs, then the expectations under the Hardy-Weinberg equation, it would suggest that that population must be evolving. So I'm going to break it down for you. There's a calculation, and in the calculation, there's expectations. And if it turns out that the answer to it differs than what the expectation is, then that population must be evolving. So with this, so we're going into what P and Q. So we have some variables, P and Q. P and Q represent the relative frequencies of two possible alleles. Okay, so we can only compare two alleles at a time. We can't, like, for example, with a frog. Well, no, I don't want to give that as an example because it has to do with what they eat. We can't compare three alleles. We can't compare in a human brown hair, blonde hair, red hair. We have to deal with two alleles in a population. Alrighty, so when we talk about and represent P and Q, P is going to represent the dominant trait. Q is going to represent the recessive trait. So let's change those names. P represents the dominant allele, Q represents the recessive allele. Okay, so this is your equation. Don't let it scare you. It's not as ugly as you think. P squared plus 2PQ plus Q squared will always equal 1. And what that means is, if you keep in mind the homozygous dominant, the homozygous dominant individuals in a population and the homozygous recessive individuals in a population, and you add up the heterozygous individuals in a population, that will always equal 100% of the population. That's exactly what that means. So, all righty. So as you can see here, I have my little video. I'm going to click on it. Now, this is going to lead you to, let's make sure that I am sharing the correct screen so you can see it. Yep. This is going to lead you to Bozeman Science. Bozeman Science, he is pretty awesome. He's got it all set up on how to calculate and do that Hardy-Weinberg equation. Since he has it down to a T with his awesome computer skills, I'm going to go ahead and let you watch his video. Whoops. Okay, I'm going to go ahead and let you watch his video for that example. So you can go ahead and pause my video if you like and click on his, and then come back to mine. Okay, stop share. Okay, so welcome back. So just on the rest of our lecture, in a population where your gametes contribute to the next generation randomly, Mendelian inheritance occurs. And so the alleles and the genotype frequencies remain constant from generation to generation. So what I'm trying to say is, if you have a population that produces gametes, eggs and sperm, you can apply this. If you have individuals that don't produce eggs and sperm and they asexually are reproducing, this does not apply. That's what that is saying. All righty, so we're moving on. At this point, we've assumed that you have watched Bozeman Science's video, that link. Okay, so natural selection, genetic drift, and gene flow. These are all three situations that can alter allele frequencies in a population. So there are three main, oops, I just said it. All right. So your natural selection is going to result in alleles being passed from one generation to the next generation. And so you're not going to have always the same proportions from one generation to the next. So for example, you're not always going to have the same amount of brunettes in a parent generation. And when you look at, not the parent, I don't want to call it that, in one generation, and then when you look at the offspring of that generation, you're not going to have the same percentages of brunettes in that generation. Okay, so they're going to differ. You're going to find sometimes that you have a greater number of dominant alleles in one generation, but in the next generation, you might have a lot less percentage of those dominant alleles. You might find it toggling back and forth as you go from generation to the next generation. At some point, you might find that you have literally zeroed yourself out, where you only get, we're in one generation, the very last one, all you have is dominant alleles. You no longer have the recessive allele in the population. Also, you might find it the other way, where you only have recessive allele in the population and no more dominant alleles in the population. And so, all righty. So you can go ahead and read your example in the book. It talks about alleles and DDT in fruit flies. So then, excuse me, let's see right here. Just wanted to make sure that we were still recording. It didn't appear that we were still recording. All righty. Make sure I'm still sharing the screen you want to see. Oh, looks like I wasn't. Good thing I checked on that. There. Now we're sharing again. So I was just talking here about genetic drift, gene flow, and such. I only went 2 slides, so it wasn't detrimental, hopefully. And so natural selection can actually cause and lead to adaptive evolution. Adaptive evolution is a process in which traits that enhance survival or reproduction will increase in frequency over time. This is very, very common. So I'm going to give you the example of cystic fibrosis. So cystic fibrosis is it is a genetic mutation where recessive individuals, persons with two recessive alleles for cystic fibrosis, they have an absence of chloride channels on their mucosal cells. And so what that causes an excess amount of mucus to basically accumulate in different mucosal cells. That can lead to, it could lead to death is what it could lead to. It could lead to breathing problems in the lungs where they cannot get air in, oxygen in, they cannot get CO2 out. That can lead to digestive issues that leads to malnutrition. So basically, any mucosal cells, it needs to have less amounts of mucus, and too much mucus can lead to death. So back to your adaptive evolution. In your adaptive evolution, I gotta close this window. These dogs are driving me nuts. I'm so sorry. Okay, in your adaptive evolution, that when we talk about cystic fibrosis, I am giving you an example of how a trait can actually be lessened in a population. So cystic fibrosis is still around, but the trait of not having cystic fibrosis is greater within a population, meaning more individuals in a population don't have cystic fibrosis than those that do have cystic fibrosis. So not having cystic fibrosis enhances their survival rate. It also enhances reproductive rate. And so with that, over time, as populations have evolved, you will find the number of individuals with cystic fibrosis are less and less. Okay, so that is in a population that does not have the medical technology that we have. So that is literally dealing in a natural setting. Okay, so I'm going to go ahead and put a period in that. Now, before I say the next piece, I want you to know I'm not unfeeling. I care about everybody, but this is just to provide you with some information and knowledge of how this works. So with that, in a natural setting, individuals with cystic fibrosis would usually die in childhood. They would not be able to breathe, they would not be able to get their nutrients, and they would die usually within their single digits. So that right there limits the ability for them to have offspring. And if they don't have offspring, they don't pass on that trait to the next generation. Okay, so that is in a natural setting. Okay, so now we live in an age with lots of medical technology, and that medical technology is helping those individuals with cystic fibrosis live longer, living into reproductive ages, living into early 30s. And so that is increasing their chances of survival in a longer setting. And then also that ability to reproduce and pass on those genes. And so with that, by them passing on those genes, that keeps that deleterious gene in the population in a higher percentage than what would have taken place naturally. So like I said, as I tell you this, I'm just giving the information. I never, if I had a child with cystic fibrosis, absolutely, I would want medical technology to keep them alive as long as possible until possibly they can find a cure for it. So I hope that lands well with everybody. All right, so next piece, genetic drift. What we've been talking about is leading to genetic drift. And so I'm going to skip to the definition. Genetic drift is going to describe how alleles in a population are going to fluctuate unpredictably from one generation to the next. So this is something I've already stated when I was talking about hair color. If you have individuals in a population, say, for example, say you have 3/4 of the population having brown hair and 1/4 of the population having blonde hair. Okay. So I'm going to go ahead and change those to percentages because that's usually how we look at it when we're talking about genetic drift. 75% brunette, 25% blonde. In the next generation, you might find that your individuals, you got 50% brunette and 50% blonde. When you go to the next generation, you might find you have 90% brunette, 10% blonde. Go to the next generation, you might be popping back into the 50% brunette, 50% blonde. So genetic drift is describing how the allele frequencies are going to fluctuate unpredictably from generation to generation. So the thing about genetic drift, going back into the statement up here, the smaller a sample size, the greater the chance of random deviation from the predicted result. Okay. So if you have a large population, you're going to find that your populations going from one to the next will fluctuate kind of very predictably, maybe even following around that 50% line, back and forth, up and down, up and down. But when you have a smaller population, you are going to find that one of those traits is going to eventually hit 100%. And then the other trait, usually the recessive trait, is going to zero out, where in that last population, all the population has brunette and there are no blondes. Okay. So the smaller the sample size, the greater the chance of it deviating from the predicted results. So your genetic drift tends to reduce genetic variation because, like I just told you, you get a loss of alleles, a random loss of alleles. The blondes were lost out of the population. But if you have a really big population, we could still keep those recessive blonde genes, even if they're not being expressed. Okay, so let's apply that Hardy-Weinberg to this generation right here, just with the frequencies of the P. So when you're looking at genetic drift, let's go ahead and look at your generation 1 compared to generation 2 compared to generation 3. Your generation 1, if you look at the frequencies of the color red alleles, CR, this is a specific flower that if you have 2 red alleles, it's a red flower. If you have 2 white alleles, it's a white flower. If you had a red and a white allele, it's a pink flower. This is a case of codominance. This is something that you learned in your 111. So with that, the frequency of the red allele, all you do is count it. You go in 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? And then get my pink ones, 11, 12, 13, 14. Hopefully I counted that right. Now I need to feel like I need to count again. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Perfect. Write that 14 down. So there are, actually, I will write it down for you. Let's get my beautiful pen color right here. Perfect. There are 14 out of, we don't know yet, but we're going to say there are 14 out of blankety blank CR alleles. I'm going to write CR over here. I didn't quite leave myself room. And the CW allele. Let's see how many CW alleles we have. We have 1, 2. That's it for the white. Okay, let's go to the pink. 3, 4, 5, 6. So you have 6. So in this population, there's a total of 20 alleles. 14 out of 20 are red alleles. 6 out of 20 are white alleles. So that's where they're going to go ahead and figure out the frequency. So just go ahead and pick up your calculator. I will do the same right now. 14 divided by 20, look at that, 0.7. That's where they get the 0.7 from right there. And then go ahead and do the math if you like. 6 divided by 20 is 0.3. And so that's where you get your 0.3 right here. But guess what? You really didn't have to do the math. If this is 100% population, it's a whole population. If you know how many red alleles there are, 0.7, well, take 1 minus 0.7 and you get 0.3. So if you recall on those, they're always going to equal 1. And 1 means it's 100% of the population. So of this generation 1 population, you end up with 5 plants leaving offspring. And these are the resulting offspring right here. So now the generation 2 population, the allele frequency has changed. So P is representative of the red allele, the dominant one. Well, in this case, it's not dominant because these are codominants. And then the Q represents of the other allele. Like I said in the previous one, it's usually the dominant, but it's not always the case. So now you have a frequency of 0.5. So 50% of the population is carrying red alleles. This is not telling you what the combination is. You don't know that it's a red flower. You don't know that it's a white, well, you would know that there's some white flowers. Well, maybe not. It could be that you put them all together and they're all heterozygous. So to look at this doesn't tell you the color of the flowers. To look at this tells you the population, 50% of the alleles of the population are red. 50% of the alleles of the population are white. Alrighty, so of this population, 2 plants leave offspring. Then we end up with all red alleles. So this is that case where I was telling you that it could zero out. 100% or 1.0 of the population are red alleles, and 0%, which is 0.0 of the population are white alleles. We now have no white alleles in this population. So this is an example of a population when it is a smaller population where you can lose alleles out of the population. So an application of genetic drift is called the founder effect. And this is a very specific application of it. And so the founder effect is going to occur when a few individuals become isolated from a larger population. And so this right here, the fact that only 2 plants left offspring, as an ecologist, I would be like, why only 2 plants? What's wrong? Did they get isolated somehow? Did something happen to the area that only these 2 plants could get together? And so the founder effect occurs when you have an isolated population and then only a few individuals are there to make the babies. So in that founder effect, allele frequencies in smaller populations can be different than the larger parent, meaning that if you just have a couple individuals that go off and go on an island together, and they are now the new baby makers of the island, you're going to end up with way different allele frequencies than the population of the mainland that had lots of different individuals. The bottleneck effect. The bottleneck effect is another special application of genetic drift. The bottleneck effect occurs when there is a drastic reduction in the population size due to a sudden change of environment. Okay, so when we're talking about the founder effect, that is an isolation of individuals from a larger population. The bottleneck effect is when there's some type of environmental change that basically wiped out a lot of individuals. leaving a smaller number of individuals to start the new population again. And so the result of the gene pool may no longer reflect the original population's gene pool. Now, if the population remains small, it may be further affected by additional genetic drift. So when you look at your bottleneck effect, let's go ahead and look at the original population. So here is your original population. You can see there's a lot of blue alleles. There's a lot of white alleles, okay? And there are very few yellow alleles, right? So if you want to go ahead and put some value to this, we're going to say brunettes, blondes, and redheads. All righty. So as your original population, say we're going to go ahead and say a tornado comes through this town, and the tornado wipes out a bunch of these individuals, and only a few individuals survive. Okay, so let's go ahead and shake out those individuals, leaving just a few alleles. So we shake them out, and this is now the alleles of the surviving population. Well, it looks like the surviving population doesn't get any redhead alleles. So that means future populations, that redhead gene is gone for future populations. You can also see that the blonde allele, it's very limited. But these are the alleles that are going to be left to make further generations. So the bottleneck effect can reduce the amount of genetic diversity within a population. So understanding this bottleneck effect can increase the understanding of how human activity affects other species. And so this talks about the prairie chicken, and this goes into the study of the prairie chicken and how the bottlenecking effect has affected the prairie chicken. And so when you look here, here's your pre-bottleneck effect, and here's your post-bottleneck effect. This is in Illinois. The range of the prairie chicken was very, very wide, lots in Illinois. This is a population study from 1820, and this is a population study from 1993. So if you look down here in the chart, in the table, from 1930 to 1960, you had a population size of 1,000 to 25,000 prairie chickens, okay? These are actual individuals. So when we're talking about the alleles that those individuals are carrying, the number of alleles per locus, so location on their genes. So they had about 5.2 alleles per locus, okay? And this is telling you about how many eggs are hatching. Those individuals that hatched eggs, there were 93 eggs hatching total. If you want to compare it, 1993, population size in Illinois, less than 50 chickens. Literally from this time frame of 1930 to 1960, we had anywhere between 1,000 to 25,000 chickens. Now we have less than 50. And this shows your allele frequency has gone down to 3.7 alleles. With that, you are seeing less chickens, less eggs being hatched. So this is showing you that because you have less genetic diversity, i.e. alleles, you are actually having less viable offspring. So versus the generations in the 1930s to 60s. We want to compare it to Kansas in 1998. Kansas did not have a bottlenecking effect, and they had 750,000 prairie chickens, 5.8 alleles, and 99 eggs hatched during that year. 99% of the eggs hatched that year, meaning that genetic diversity of having lots of alleles was a positive effect on the ability for viable offspring. In Nebraska, same thing, 75,000 to 200,000, no bottlenecking took place there, a greater number of alleles and a greater number of percentage of eggs hatching. I'll let you go ahead and read through this study if you like. This is just a summary of genetic drift. So genetic drift can be very significant in small populations. Genetic drift can cause allele frequencies to change at random. Genetic drift can also lead to a loss of genetic variation within populations, and genetic drift can be cause harmful alleles to be fixed within a population. So this is basically a summary of everything what we just talked about with those key points. So let's talk about some gene flow. Gene flow is going to consist of the movement of alleles among populations. And so it sounds exactly like it is. The genes are going to flow within the population. And so alleles can be transferred through the movement of fertile individuals or gametes. For example, pollen. Pollen is an example of gametes being moved from maybe one population to another or within a population. And so pollen is the gamete of the male angiosperm. And so angiosperm, most flowers, okay? And so pollen can move in several different ways. Pollen can move via wind. You know, when you see a dandelion and you see the pollen just flowing through the air of the dandelion, pollen can move via insects. Insects rub their body on the flowers, and then they move on to a new flower and they dust off the pollen onto a new flower. So the pollen is going to be the male gamete of your plants. And so gene flow tends to reduce variation among populations over time. And so we want gene flow within a population. That is important. Gene flow can affect the ability for a population to adapt to local environments. If you do not have enough gene flow, that can directly affect a population's ability to adapt to an environment, and that can be detrimental on a population. So, for example, the banding pattern in Lake Erie water snakes represents an adaptation to the mainland and the island habitats. So most of your snakes on the mainland are going to be very strongly banded. Most of your island snakes do not have bands, or they might have less bands, smaller bands, less of them. Your unbanded snakes are going to be better camouflaged in an island, okay? So the banding is a result of the genes. Their ability to be camouflaged within the environment, that is an adaptation. So how they adapt in their environment with their camouflage. So, but migration of the banded snakes from the mainland to the island populations, that helps to maintain the alleles within the population to keep banding patterns in the island population. Well, like I just told you up here, unbanded snakes are better camouflaged in the island. But when these banded snakes keep swimming over and mixing with the population, that is making it difficult for the unbanded snakes to continue their camouflage. Alrighty, so we're almost there. Evolution by natural selection is going to involve both chance and sorting. And so new genetic variations, they arise by chance, okay? That has to do with genetic mutation, and it's really just happenstance. And so beneficial alleles are going to be sorted and favored by natural selection. It is nature that is going to select which phenotypes are going to be the most suited for the populations. Well, the phenotypes are a result of the alleles, i.e., what causes the genotypes. So only natural selection consistently increases the frequencies of alleles that provide reproductive advantage. What this means is if you got good alleles, that are, quote, good for the environment, meaning you are successful in the environment you are. You have a reproductive advantage. We've kind of talked about this a lot, so I'm not going to continue. So with that, leading to what is called the relative fitness. I'm going to go ahead and give me a second, escape out. We're on 43 out of 56. We're getting there. We're getting there, guys. I know you're probably wondering how much longer. So your relative fitness. That phrase, basically, the phrases struggle for existence and survival of the fittest. They're kind of misleading. They imply actual competition directly between individuals, okay? Really, it's about that individual's ability to live in a population and be successful. So your reproductive success is generally more subtle and depends on many different factors. And so when we talk about the term relative fitness is the contribution an individual makes to a gene pool of the next generation relative to other individuals' contributions. So selection is going to favor certain genotypes by acting upon the phenotypes. So if for this stuff I already said, for example, if my alleles add to the gene pool a very favorable alleles for that environment, you can say that me as an individual, I have high relative fitness versus maybe my neighbor doesn't have such favorable alleles. His relative fitness is low in comparison to mine. So there are going to be 3 different models of selection. You should know these for your exam, definitely. I mean, you should know all of this for your exam, or else I couldn't keep repeating the same things over and over, because the book does, by the way. Directional, disruptive, and stabilizing selection. So directional selection is going to favor individuals at one extreme or the other. Okay, I'm going to show you some pictures here in a moment. Disruptive selection are going to favor individuals at both extremes, but not in the middle. And stabilizing selection is going to favor the intermediate of the extremes, but not the extremes themselves. So when you look at your model of your mouse, this is a population of mice. They are of the same species, but they just have different alleles of fur color. Okay, just like we're blondes and brunettes and redheads, these mice are white alleles and light brown, medium brown, dark brown, and black. So with that, in this population, you are going to find individuals in that population with all of the different fur colors. And what this is showing here is the number of individuals with this, the frequency of these alleles, meaning you have the greater number of individuals being medium brown, less individuals being white, less individuals being black, and then kind of moderate on those moderate phenotypes of fur color. All right, so when we're talking about directional selection, if you look here, here is your original population. What directional selection does, that will literally push the population in a specific direction, meaning next populations will push in this, where's my, oops, I'm sorry, I'm trying to write, because I'm not connected with my pen. There we go. Push in this direction towards these phenotypes. Okay, so you are no longer having the white, you are no longer having the light brown, and you are now having medium brown, a little darker brown, and black. But when you look at the alleles of the population, this new population, the next generations, are having higher amount of individual alleles, or not individuals, but frequency of alleles being those that cause darker brown fur. And so we are going in this direction. That is directional selection, where it will select towards the more extreme phenotypes. All right, so disruptive selection does the difference. So your original population was the Black, and it had a higher allele frequency towards the intermediate. But in disruptive, what it does, it is going to select for the extremes of the population. But it's going to deselect for the intermediates, meaning you're going to have a lot less intermediate frequencies of allele frequency, and you're going to have a greater amount of allele frequency that are going to be the darker phenotype, or the darker alleles, I should say, and the alleles that cause the lighter phenotype. Stabilizing selection. Stabilizing selection is going to select for the intermediate. So you're going to get a lot more individuals in the population having the intermediate color, and pretty much you're going to have less or none of the extremes. You're not going to have the white, you're not going to have the black, you're going to have a lot less of the light brown, a lot less of the dark brown, and you're going to stabilize around the medium brown. So the key role of natural selection in adaptive evolution. So striking adaptations have arisen by natural selection. For example, different octopus, they can change their color rapidly for camouflage. That is an adaptation. The jaws of snakes allow them to swallow larger prey. The fact that snakes have this ligament here that allows them to open their jaw up wider, that is that adaptation that allows them to swallow prey larger than their head, that movable jawbone. And so your natural selection increases those frequencies of alleles that enhances survival and the reproduction. And so that adaptive evolution occurs as a degree to which the species is well suited. I feel that this is stuff we've already said. I'm tired of saying it. It's just a different application of it. And so because that environment can change, adaptation is continuous. That adaptive evolution is continuous. And so your genetic drift and your gene flow, they do not consistently increase the frequencies of alleles that enhance reproduction, meaning it is chance. It's not about the enhancement. They're not selected on because those alleles enhance their survival. Your genetic drift and your gene flow are more random, as stated previous. Both of those processes may increase or decrease the frequency of the beneficial alleles in the population, meaning you could end up with more deleterious genes, or you can end up with more beneficial genes. The term deleterious means it is not in that organism's best interest, and it will probably cause it to not survive. That's what deleterious means. All righty, sexual selection. It is the process in which individuals with certain inherited characteristics are more likely to acquire mates than their individuals of the same sex. And so before we talk about that, let's talk about the term sexual dimorphism. Sexual dimorphism is a very obvious difference between the genders of an organism, okay, between the sexes, between the male and the female. Some organisms aren't just about male and female, but for the most part, that's what we think about. So as you look here and you look at your peacock, the peacock that you see, this is the male peacock. And the peacock over here, this is your female peacock. They are sexually dimorphic, meaning they don't look the same at all. They have big difference in their sexual characteristics. And so your female peacock, she doesn't need all the fancy feathers. Why? Because she gets to choose. She gets to select. And that's what takes us to the sexual selection. She gets to look, she gets to walk up and down the area, look at all the guys, gets to see who's got the prettiest fan. Okay. Not only who's got the prettiest fan, who has the fullest fan, who can shake their fan the best. Notice how peacocks will walk up and they'll shake their fan at the girl. Well, who could shake it the best? She gets to decide. And because of that, that leads to sexual selection. So if you had a male peacock that, we'll say that maybe he hasn't developed a great fan yet, well, she's not going to choose him. He's not ready. Okay. He's not going to be a good father as far as she's concerned. What she sees, he's not offering good genes. And so also it could be a matter of nutrition. And so if she sees the male and his feathers aren't as vibrant, his feathers aren't as filled in, he doesn't have a full fan, his fan is shorter, she might see that as this is not a good choice in mate, because this individual, he doesn't look good enough. Now, which also leads to another thing. The bigger the fan, the more likely you are to get taken up by predator, right? That fan is it's a pain in the butt to drag around. Well, if you're still sticking around and you got a big fan and you're still sticking around, that female peacock sees you as a catch. They see you as, oh my gosh, they have that huge fan and they're still around and evading predators. He's got some good genes. That's the one that I want to mate with. Okay. So an example with sexual dimorphism, let's go ahead and say in humans. Humans are sexually dimorphic. If you look at the male of a species and the female of the species, not only just looking at the obvious, the sexual organs, females having breasts, males having a penis, that's more of the obvious thing. But that sexual dimorphism, if you look at a male and the size of a male's chest due to the increases of testosterone, and I'm not talking muscles, I'm talking about bone structure. I'm talking about the width of a male human's chest in comparison to the width of a female human's chest. Because of the testosterone, that causes them to thicken their bone structure and widen their clavicle. And so that is a perfect example of sexual dimorphism. Also height, sexual dimorphism. Another one, facial hair, sexual dimorphism. So all of those examples, you can probably think of more. So when we talk about intrasexual selection versus intersexual selection, intra means within, okay? Intra is a direct competition among individuals of one sex, the males. Intrasexual selection, the males are one sex for mates. Basically, they're fighting. They're fighting to show who's the best. Okay. Intersexual selection, it is when the female makes the choice. The female gets to choose the mate. So if I go ahead and, I didn't have a nice picture. That's all right. I was just going to say rams. They're gonna fight to determine, or back to gorillas, of course, they get to determine who gets to be the silverback. Whoever is the most dominant, he's the one who gets to be the silverback. Intrasexual selection versus inter. Perfect example I just showed you on the previous slide would be the peacocks. And so that showiness of your male appearance, that can increase a male's chances of attracting a female, but like I said, could also decrease his chances of survival. So if he gets to mate, he must have done right. And so how do females' preferences evolve? Like I was saying before, there's the quote unquote good genes hypothesis. That suggests that if a trait is related to the male genetic quality, both the male and the female, both the male trait and the female preference for that trait will increase in frequency. So if the female keeps preferring the prettiest, she's going to keep mating with the prettiest, which means her sons are going to be the prettiest. So that means if her sons are the prettiest, some other females' daughters, they're going to have females that prefer the prettiest. So basically, the preference is for the prettiest, and the males keep getting prettier. All righty. Balancing selection. So diploidy maintains genetic variation in the form of recessive alleles hidden from the selection in heterozygotes. Okay, so heterozygotes means that there is one dominant trait and one recessive trait. The recessive trait is usually hidden. So meaning just because you look at an individual and you see their phenotype and they are expressing the dominant trait, you do not know if they are carrying the recessive allele because that recessive allele is hidden. So the balancing selection occurs when natural selection maintains stable frequencies of two or more phenotypic forms in a population. And so your balancing selection includes two things: frequency dependent selection and heterozygous advantage. So let's look at the first one, frequency dependent selection. That is, so the fitness of a phenotype depends on how common it is in the population. So if you look at these fish, here, you see this is a sucker fish. And this sucker fish's mouth leads to the left. It leans left. This is a same species of suckerfish, but he's got an alternate allele. And that alternate allele causes his sucker mouth to grow to the right. And so in this frequency dependent selection, that frequency dependent selection results in approximately equal numbers of the right-left suckerfish. So when you look here, 81, 83, 85, 1987, 1988, they did a survey. You had a lot more that were, let's see which one, I believe this one is to the left, yep, and then to the right. In this year, a lot more to the left, a lot more to the right, a lot more to the right. In the middle, 50-50, a lot more to the left. So this one, I guess, was kind of a 50-50 too. So you are going to see that frequency alternating. And it's going to continue to do that until there's an obvious advantage of being one or the other. And at the point there's an obvious advantage of being one or the other, that's when you're going to see a change. Okay. Let's go to the heterozygote advantage. Heterozygote advantage occurs when heterozygotes have a greater fitness than the homozygotes, meaning homozygous dominance or the homozygous recessive. When the heterozygous, they have the advantage. So nature is going to select to maintain two or more alleles at that locus. And so when you have a heterozygote advantage, that's going to result from stabilizing or directional selection. Perfect example here, something you've seen in Bio 111 is going to be your sickle cell disease. So sickle cell disease is going to result when you have two sickle cell alleles, and your sickle cell alleles are recessive alleles. Okay, so most people, especially on the northern hemisphere, I don't want to say the northern hemisphere, but sickle cell is something that comes from, it is a mutation. And there are great numbers of mutations in individuals that are of African American descent. Okay, so that is a mutation that is carried on through being a heterozygote. Usually an individual with sickle cell does not survive, okay? Or they have a low fitness where they do not have a long quality of life. Those individuals that are homozygous dominant for not having sickle cell, they live the normal life, not affected from the allele of having a non-sickle cell alleles. But when those individuals go into an area of malaria, an area of malaria, that's what I was trying to say. Those individuals that have dominant trait 2 alleles for non-sickle cell, they tend to get malaria easier. They, whenever they get infected with malaria, they usually die from malaria if it's not treated. Well, the sickle cell the heterozygous advantage application to the sickle cell allele is that individuals with one allele for a normal red blood cell and one allele for a sickle cell shaped red blood cell, they're heterozygotes. But because those heterozygotes, if they are put in an area of malaria, they tend to be resistant to malaria. They have a lower fitness of life, meaning that they still suffer from some sickle cell symptoms, but it's not that bad that it is fatal. So it just lowers their fitness in life. So that is the heterozygote advantage. The fact that a heterozygotes, though that person that is carrying the sickle cell allele, but also has a dominant allele, they have an advantage in an area, in an environment that has high malaria. They are resistant to that malaria disease. All righty. I hope that was helpful. Let me know if you have any questions.